Welcome everyone. Today we're doing a CAPE Biology past paper review. We're doing a Unit 1 paper and we're doing a paper 2. This was from the year 2019, and please to note that there are 3 questions on this paper, each worth 30 marks. I'm only going to do question number 1 today, and the point to note is that question number 1 is from module 1, question number 2 is from module 2, question number 3 is from module 3. Again, I will come back and do the other questions. So let's start off with question number 1, but please also remember, if you start the examination, to read through the instructions carefully, and if you're in doubt, please ask questions before the examination begins. Now for question number 1, which is from Cell and Molecular Biology, again, it is from module 1. And so the question reads that figure 1 shows a light micrograph of a transverse section to the primary stem of a dahlia, and is a dahlia species plant, which is a typical dicotyledon. Now what I'm going to do first here is to label these structures. All right, so let's start off with labeling first, and so I'm going to label structure A, which is the epidermis. And then let's start with structure B, and structure B here, it is the cortex. Structure C, that is called the pith. Structure D, that is the xylem, and is the xylem vessel. Structure E is the phloem. All right, and so we have more structures which I'm going to label as well for you. And so let's look at a few other structures that we can talk about. Let's put them on this side. So I'm going to label this region here that separates the xylem and the phloem. So I'm going to talk about this layer. I'm just put a marking right there. Okay. And so that layer that separates the xylem from the phloem, I'll label that part. And I'm also going to label this part right here that is not labeled. And I'm going to label this region right here as well, which is on top of the xylem and phloem, which is on top of the vascular bundle. So I'm going to label those 3 parts. No, I'm going to label another part right here as well, this little part right here. All right, so I'm going to label 4 parts. All right, so let's start off with this one here. And it's a layer just below the epidermis, and that layer below the epidermis is called a collenchyma. So here is called a collenchyma. And the collenchyma of the below the epidermis, or of the cortex, and the portion that separates the xylem from the phloem, this is called the cambium. So the cambium separates the phloem vessel from the xylem vessel. And then here we have the endodermis, which is the innermost region of the cortex. And then we have right here, it's called a collenchyma, which is the top of the vascular bundle. So it is a vascular bundle collenchyma. All right, so sclerenchyma of the vascular bundle. All right, so those are the parts that I want you at least to know. All right, so what we're going to do now is to jump into the question itself to see what is being asked. Okay, just make a note of these parts or structures. All right, so the first part of the question now, it reads that in the space below, construct a tissue diagram from the light micrograph of the plant section. So what plant section they're talking about here, it is the stem of the dicotyledon plant. So we can always go back to the plant to see, or to the diagram to see the structure here, right? All right, so what we're going to do is to consider what is a plant tissue diagram. And so if you think about the word tissue, we're talking about the section of a plant that contains cells or made up of different cells. But what I want to make reference to before I even start the question, I'm going to make reference to it at the top of this, just to go a little bit up. All right, I want to just give you a little bit of extra information right here. And so what I want to make reference to in terms of organs, we have some organs of the plant. There are three major organs of the plant, right? Just to point it out. The organs of a plant will include the root, the stem, and the leaves. All right, so those are the different organs of the plant, okay? All right, and so in terms of tissue are grouped in different categories based on their function as well. So in terms of plant tissue, we'll have the vascular tissue, just to make a note of that. We have vascular tissue. We also have the epidermal tissue. All right, so we have epidermal tissue. We also have the ground tissue as well. All right, and we have also the meristem. All right, so those are generally the different types of tissues that we'll have in a plant. All right, so now what we're going to do now is to go into the diagram of drawing a tissue diagram. And before I even start the tissue diagram, what I want to focus on is the vascular tissue. So I'm looking at the vascular tissue first. All right, and then I'm going to zoom into it to draw a specific tissue itself. So first, let's start with the vascular bundle. All right, I just want to draw this for you to see it and label it properly. So the vascular bundle, it has this type of shape here. All right, not perfect, but yep. And then on the top region, we have a little section right there. And then let me use a thicker line to kind of separate this. All right, I'm going to do that. Awesome. All right. So what I'm going to do is to label these parts. And this is not the tissue diagram I'm actually drawing. This is just the vascular tissue. And then I'm going to get into a specific part. All right, so that is one section. That's another section. And this is another section. And what I want you to do is able to identify these parts of the vascular bundle. All right. And so on top, which I labeled these in the diagrams before, but I just want to point them out right here. So the uppermost part right here is called this Collenchyma. This is the Collenchyma of the vascular bundle. And then on the top part of it, we'll have the phloem. And I'm going to go back to the diagram to show you something before I actually draw a tissue diagram on the side of this. And then we're going to have here the cambium. The cambium is what separates the xylem from the phloem. And then we have the xylem on the innermost part. All right, great. Now, what I want to ask you though, or for you to consider, is why the xylem is the inner part of the vascular bundle and the phloem is at the outermost part in the stem of a dicotyledon plant. There is an adaptation for that. So I want to show you on the diagram itself. So if you notice right here, you'll realize that the phloem, right, let me just kind of highlight that. So the phloem is this portion right here, all right, in yellow. And so let's now look at the xylem, which is the inner part. So notice xylem here is the inner part, and put that in red. That's shaded a little bit in red. So notice where they are located, and there's a reason for that. The phloem is outermost of the vascular bundle in the stem of a plant, a matter of fact. And there's a reason, because the phloem can be more exposed to the external environment. If the xylem was on the outer part, then the xylem would lose water by evaporation or transpiration on the sides of the plant. So the plant stem is adapted to make sure the xylem is on the innermost part to reduce water loss by evaporation or transpiration from the sides of the plant. So the phloem now will kind of create a shield towards the xylem. All right, so the xylem is more protected from the external environment. So instead of water moving to the sides of a plant, water will go straight up into the leaves of a plant where they're most needed. All right, just to point it out. All right, now let's go back to the question and do some more drawing right here. All right, so what I want to draw is the phloem vessel for you. I could draw the xylem, but the xylem is made up of many dead cells. But I want to do something that contains more structures, and I'm also going to label that. So in terms of the phloem, which I'm drawing right now, we're going to label that as well. And so the phloem will be made up of different parts and different cells, different type of cells. I'm going to put some broken line right there to present my sieve plate, and my sieve pores will be through those broken lines. And what interestingly I want to put on the side right here is a specialized type of cell. All right, so let's put them right there. And there's another one will be up here as well. So I'm going to give some labelings to identify what I'm drawing actually. Because the diagram should always be annotated. Please always try to do that as best as possible. Annotate your diagrams. All right, so I'm going to label this portion right here. So let's put an arrow right there. I'm also going to label this cell, which is very important. I'm also going to label this part. And then I'm going to give a name to the entire region as well. All right, so let's label this. So this here is called a sieve plate. This is our sieve plate. Here is what they call a sieve tube. And this is what they call a companion cell. All right, but what I also want to point out is that the region where the sieve tube is the sieve plate and the companion cell is actually called the sieve tube element. So all of this region right here, I'll put it at the bottom. So this entire region, let's put this in the blue. From here, coming down, all of this region is called the sieve tube element. So they're all together sieve tube element. All right, so now you have that. So this tissue is made up of many different types of cells. And just to make a note that this is a phloem, and the phloem, just pretty label that as well. So please also label. So this is a phloem. All right, and the phloem is responsible to transport plant food in the form of sugars. So sucrose will be transported, and the process by which a phloem transport plant food is called translocation. Just to make a note of that as well. All right, let's go to the next part of the question right here. And that's a 3-mark question. Here it said provide annotations to describe each labeled structure A to E. All right, and so let's quickly talk about them real quick. And so A, as I remember, that is epidermis. So you could always go back to the diagram and you will check them out and see the labelings that I labeled there. So A is the epidermis. Okay, B is the cortex, C is the pit, D is the xylem, E is the phloem. All right, so you can remember that epidermis, cortex, pit, xylem, and phloem. All right, so let's go to the epidermis first. And so when we talk about the epidermis is a single layer of cell that covers the cell. So it is a single layer of cells that cover the plant stem. All right. And very important to note that this serves as a protection. So this serves as protection for the plant stem. All right, great. And now for structure B, as remember that again is a cortex. So structure B is a cortex. So let's talk about the cortex. And the cortex is found in terms of describing the cortex is actually is a number of cells, really is a greater number of cells that are there. And these cells are actually separated. So the cortex now is found between two different layers or two different structures. It's found between the epidermis and the vascular bundle or the vascular tube. So the vascular bundle or vascular tube or vessel, let me say vascular vessel. All right. All right, so that's where it is found. And what is important about the cortex is that the cortex will help in terms of transport and support. All right, and it's also help with protection as well. So the cortex helps with transport. All right, transport, support, and protection. All right, just to point out those things about them. And then C, which is the pith. So the pith, and the pith is soft in texture. So it is the soft innermost, let's say soft innermost layer of the stem. All right, so the soft innermost layer of the stem. But what is it important for? It's very important in terms of transport. Someone says aids in transport and storage of nutrients or minerals, right? So that's very important right there. So importance of storage and transport of nutrients. All right, and so let's go to the next structure now. And D is the xylem. And just to make a mention right here that the cortex and the pith, they contain parenchyma cells, just to make a note of that both cortex and pith, they contain parenchyma cells, so you can make a note of that as well. Now, the xylem, which is made of parenchyma and what they call tracheids, I'm going to talk about tracheids as well. So the xylem, it is really a layer of dead cells or tissue of dead cells. So I'm going to describe it, say it contains or made up of dead cell, contains dead cells. And these dead cells, they are called tracheids, just to make a mention of that. They're called tracheids. All right. And the function of the xylem is to transport water and mineral salt. But let me give another description right here in terms of the xylem. The xylem is also long, narrow tubes, right? And the long, narrow tubes are made up of two things. They're made up of the tracheids and also what they call the vessels. All right? The vessels, they are the tube-like structures that connect end to ends, while the tracheids, they are narrow, long, continuous tubes. All right, and these are very important because they are dead. Water could pass through them easily rather than being absorbed by them. Okay, so they help with capillary actions of water, the movement of water. All right, so again, just to make a note that they are long. Xylem is long, so we say xylem is long, narrow tube. All right, and let's just say used to transport water that aids in the transportation of water and mineral salts. All right, just to do that. All right, great. All right, so the next structure here now, which is the last structure, is the phloem. And the phloem, which is different from the xylem in terms of living and dead, because the xylem is dead, the phloem is living. So I'm going to say here now that the phloem, all right, the phloem is a living vascular tissue, right? the living vascular tissue. All right, and made up of many different cells. That is made up of many different cells, such as we talk about that in the diagram that I drew earlier, such as the sieve tube. We have a sieve tube, or the sieve element. That's the sieve element to make it better. So made up of a sieve element, which contains the sieve tube, the sieve plate, and the companion cell. All right, so it's the sieve element is there. All right, we also have parenchyma and sclerenchyma, just to make a note of that. That's why it's living. So made up of sieve element, it made up of the parenchyma and also sclerenchyma. All right. All right, so very important with that. And also, if you want to add this, it helps in transporting, so it transports plant food. Okay, so transport plant food. All right, and they transport plant food in the form of sugars, and the sugar is sucrose, and the process by which food is being transported through the phloem vessel is called translocation. All right, so now let's go to the next part of the question here. This question here, this part B, it said the graph in figure 2 shows the change in the rate of uptake of two substances, substance A and substance B, as concentration of each substance increases across the lipid bilayer. And when we talk about lipid bilayer, we're talking about a plasma membrane. Now, firstly, let's observe a graph and look at what the graph is saying. Now, you have rate of diffusion, which means the rate of which the molecules move across the plasma membrane or the lipid bilayer. So, in moving from an area where there's a certain number of molecules, I'm not going to talk about concentration yet until I actually get into it. But what I wanted to realize here is that the concentration of solute is increasing along the x-axis. I notice the different changes in the graph. Graph A is going straight up like that. So as concentration increases, the rate of diffusion increases. Graph B, it goes up. That means there was an increase, and then the graph start to plateau. Now, let's see exactly what they're asking. But remember now, diffusion is where particles will move from high concentration to low concentration. That's a general movement. All right, so that is down a concentration gradient. However, if it is the reverse is happening from low to high, then we have what they call active transport. All right, so I just want to mention that before I get into the question. And so, just a statement, it's a figure 2, graph of change in rate of substance uptake. So part 1 of this question is asking us to describe the changes in the rate of uptake of substance A and substance B as concentration of solute increases as depicted in figure 2. So looking at the graph carefully, as I mentioned again, you will notice that substance A will be in a straight line, right? And of course, you will say this is proportional graph, right? While graph B will go up and then it start to plateau. All right, so now let's talk about describing the changes here. So for graph A, so let's talk about graph A first. All right, and what I want to point out is a few things on the graph, a matter of fact, before I actually go into this. Let's just quickly see if I could do that real quick. All right. And so what I wanted to notice that this line, if they are different, there are two different things taking place right here. So this line, I'm going to label it as simple diffusion, right? Got a simple diffusion taking place right there. And this one is called facilitated diffusion. All right. And so what I want to make a note right here in that for A, which is the simple diffusion, let's put A, when I'm talking about graph A, it depends upon the concentration. So the rate depends on concentration, all right? So this depends on concentration. All right, so it depends on concentration of the solute or the substance. All right, and so, and B now, which is a facilitated diffusion, it depends upon the number of proteins channels. So this one depends on the number. Let's put number, the sign as short. The number of, all right, let me not say anyway. Let's write out all of that. The number of protein channels. All right, so this is very important, right? It doesn't make a note right there, so at least when we start our discussion, we can actually refer to this, okay? All right, so now they ask us here in the first part to describe the changes. So in graph A, all right, or for substance A, so graph A, and in graph A, what taking place right here is that as concentration increases, so as concentration increases, right, the rate of uptake increased. All right, very important to note. All right. All right, so great. Now, just to point out, to make a note right here that this is a linear change. That this, I can say the rate is linear. All right, so therefore mean it's on a linear, that means it's on a straight line. And of course, they are also directly proportional. Let's not use it directly, but they're proportional. All right, even though they start through the origin, but let's use the rate proportional to be safe right there. All right, so as rate and concentration are proportional, right? So let's make a note of that. So rate of uptake and concentration are proportional. All right, just to give a little bit of extra right there. Now for graph B, all right, in terms of description of the changes, so graph B, let's put graph B right here. And this is a 2-mark question, right? So it's not a lot of stuff that you necessarily need. But then just explain what this graph is actually showing. So in graph B now, as concentration increases, all right, the rate there, let's say this, answer this, there is an increase. There is an increase in the rate of uptake. Then the rate will plateau. All right, so which simply means now that after a period of time, when you reach that maximum amount that can go in at a particular time, it maintains that maximum rate. Because again, remember, it depends on the channel. So it depends on the channel, the amount of channel present. If you only have 3 channels, for example, the 3 channels can only take in so much and no more at a particular time, right? But in the simple diffusion, so I'm going to talk about that. It is going directly through the lipid membrane, so therefore it's not depending upon the structure. It just goes straight through it. And so as concentration increases, as much could go in as possible. All right. All right, so here now we say identify the types of diffusion illustrated by substance A and substance B. And so we talk about that on the graph itself. We talk about simple and facilitated. So let's put it right here. So for graph A or substance A, it is simple diffusion. Okay. All right, and for B, it is facilitated. So B will be facilitated diffusion. All right, so awesome. All right, so here now, in part 3, it's a compare and contrast the 2 types of diffusion identified in B2. Now, for this, what I'd like to do, instead of writing a whole lot of things, right, and to write in sentence and paragraphs, sometimes information get lost when you look at comparing and contrasting, right? And so you can look at the similarities and differences. That's what they're actually asking you to look at. Okay, so you can look at what is different, what is similar between them, and so on. But how I like to do this in examination setting is to draw a line straight through. So I'm going to leave it right here so I could, perfect. All right, so you're right there, I want it. So what I'm going to do is to draw a straight line. You can use a ruler to do this. Of course, I'm not going to be perfectly straight, because my hand is not straight. But anyhow, something like that, right? So let's call that a straight line. So on this side here, I'm going to put my simple diffusion on this side here. All right, let me just put this in red so you can see the simple diffusion here. And over this side, I'm going to put facilitated diffusion. All right, and so what I'm going to do now is to talk about the differences and similarities here. So let's put that in blue now, right? Just so we see the difference. You actually could number these. It's best to actually number them. So I'm going to number them. Let's call this 1. I'm going to compare the first thing and contrast and so on, right? So the first thing about these is that no energy is required, right? So no energy is required for both of them, all right? All right, and here I'm going to say again, no energy is required. So that's number 1, no energy. And the reason why I want to do this is also to point out something to you, is that when you're comparing and contrasting, please look at the same properties or the same structures, right, or the same characteristics. If I'm talking about energy, talk about energy for both at the same time. Don't talk about energy for one and talk about movement for something. Don't do that. Always look at the same factor or variable at the same time. Let's go to 2. The important thing right here in 2 is that the next important thing for both of them is to talk about the movement of substances. And so substance move down concentration gradient, which means from high to low concentration. So movement is down the concentration gradient. All right, I'm going to do this. Let me just go back here. The movement is down the concentration gradient. All right. And the same thing as well for the facilitated diffusion is that, let me say, down concentration gradient for short. All right, the down concentration gradient. And again, what this means is that the substance will move from high concentration to low concentration, right? And so we can also say that this is passive because there's no need for energy. So if no energy is required, then this is passive. Let me just put passive right here just to kind of make a note of that. So this is a passive form of movement, okay, a passive transport. All right, so it is passive, no requirement of energy. The third thing I can talk about here is a 4 marks question. They're looking at least 4 things, 2 differences, 2 similarities. I could mix them up, don't matter. But it's a compare and contrast. So I'm going to give you more than 4 things though. All right, so next thing we can talk about is that movement is, in terms of movement of particles, in simple diffusion, they will go straight through the membrane, okay? All right, so substance move, let me see. Substances move directly through the membrane. And this is phospholipid membrane. Okay, that's what the question actually was saying. So through the membrane, I'm not going to say through the membrane, but you can also put the phospholipid membrane. All right. But in terms of facilitated diffusion, now the substance don't move through the membrane, move through what they call it channel proteins, right? So the substance now is going through channel. Let me just put substance here. Substances move through channel proteins. All right, so great. All right, so let's look at the 4th thing about them now here. And the 4th thing we can talk about is that the substance that are moving, they're generally simple molecules. Okay, so simple substance move through these. So the substances, they are normally simple. Are simple. And when we talk about simple substances, we're talking about like carbon dioxide and oxygen, so like the normal gases really. The simple small like gases are simple and small, simple small. Or let's just say slash small. All right, so that's the general thing. And so example of this will be our carbon dioxide. So example of this could be CO2. All right. Let me write in my CO2 right here. There could be CO2, or it could also be oxygen gas as well. Okay. So that's because oxygen, O2 as well. All right. So those are the things that normally go through simple diffusion. Simple substances, simple diffusion, right? But when we talk about now facilitated diffusion now, you have more complex substances, right? So the substances are more complex. I'm going to say more complex substances will pass through. All right, and example of these complex substances I'm talking about things like glucose. All right, I'm talking about like amino acids, sugars generally. All right, so anything like that. All right, so amino acids. Let's put amino acid there since I have the space. Amino acids, yeah. So those are substances that will pass through membranes. So think about even substances that are absorbed by the cells, or substance needed by the cells, substance need to leave the cells. You can think about those substances that go in, like glucose, amino acids, and so on, right? And what is needed by a cell also is oxygen. So oxygen will diffuse in, carbon dioxide diffuse out of cells. All right. So we can look at the next one here, which is the fifth one. All right. Don't have much space. I'm going to do one more thing after this. So in terms of simple diffusion, it's generally slower. And I'm going to go on the graph to actually show you, right? So generally slower compared to facilitated. Facilitated is faster. All right. If something is facilitating, then yeah, generally faster. So this is slow, this is faster. All right. And let's put the last one right here now. The substance not bind with the membrane itself. So the substance do not bind with the membrane. Okay, so there is no binding of it, the substance with the membrane. But here now in facilitated, there is a binding of the substance to the channel proteins, right? So the substance binds to the channel protein. Okay. All right. So just to make a note of that. All right. As I was talking about faster and slower, right? I could go to the graph and show you what I'm talking about. One is fast, one is slower. Typically, again, simply slower. So notice even for the same concentration, the rate is faster for facilitated. The only time that the simple exceed the facilitated one is when the facilitated diffusion reaches its peak. Yeah, that's the only time that simple will go at a faster rate. But once that plateau is not reached, then facilitated diffusion is generally faster. Notice the line is above the simple. All right, just to point that out real quick. All right, let's go to the next question right here now. This question, with this part 4 of this question. Now, in part 4 of this question, is arrange the following molecules according to their ability to diffuse through lipid bilayer. And we have carbon dioxide, glucose, water, and RNA. So we have 4 substances. All right. So, all right. So here now, it said begin with the molecules that diffuse is the easiest. We're going to start from easiest to the most difficult and explain the order of your arrangement. So what I'm going to do is to give you the arrangement first, and then I jump into why one will diffuse faster than the other one. But you know what? Before I go into that, let me just talk about it right here. Right, there are 3 major things, three general things really that cause substances to diffuse faster across a membrane. it's based upon size. So size is important. The size is important. What is also important here as well is the solubility within that the substance. So what I mean by solubility, remember the membrane is made up of phospholipid bilayer, which is fat molecule. And so if there's fat molecules present in the lipid bilayer, then how the substance can dissolve in oil will determine how fast it will diffuse. So just to make a note that smaller size tend to diffuse faster than larger molecules. So smaller molecules tend to diffuse faster than molecules. Gases also diffuse faster than liquid or solid, right? So just to make note of that. And there's a typical reason behind that, because if gas has greater kinetic energy, then they move faster typically by themselves, right? So they can diffuse faster. Liquid has less kinetic energy compared to gas, and solid has less kinetic energy compared to liquid. So in terms of size and their movement, in terms of their kinetic energy of the molecules, determine how fast they will diffuse, right? Generally. All right, and so the solubility as well. Solubility also important in terms of diffusion. All right, and I will also say polarity. Polarity or the charge, right? So polarity is also important. Polarity or charge, all right, is very important in terms of how the substance will go across the membrane. Because remember now, the phospholipid has two parts. It has a phosphate head, which is polar, and the fatty acid tails, which are non-polar. And so based upon the polarity of the phospholipid themselves, if substances are polar or non-polar, will be affected in which or they transport through the membrane. So these are generally three things that affect the rate of diffusion across a membrane, right? And again, we can talk about size and movement of the particles and all that. So the order is going to be carbon dioxide. So I'm gonna let's see if I could put. I'll put an arrow first, and then I put my carbon dioxide just to make it easier for me to write in. All right, and then I'm gonna make a next space, and I put water there. So I'm gonna put an arrow right there, and then the next one will be glucose. I could actually type that one in directly. So glucose will be next. All right. So glucose will be next, and then after glucose will come our what was there? RNA. No, we have water. Okay, we have water, glucose, RNA. So RNA will be the last one. All right, great. So the first one will be carbon dioxide. And I know for sure there's some question behind this, while carbon dioxide will first. I'm sure you're going to ask me that. Why carbon dioxide is first? I'm going to explain it, matter of fact. So, and I hope you'll understand when I explain it. And then water will be second, because if you notice the note I make on the side, that's where the question will come from. So based on what I'm saying, then why water is after carbon dioxide. But there is kind of a dual thing. So the size is important, the solubility is important, right? But one precedes the other. All right, so I'm going to talk about that. All right, so let's jump into it, yeah? And so in terms of size, remember that size and solubility, right? Polarity also affect the rate of diffusion. That's very important to note. So why carbon dioxide diffuse faster than water? In fact, let me point this out. Carbon dioxide is larger, right? Carbon dioxide is larger than water, right? So therefore, water is expected to diffuse faster. But there's a key important thing. But matter of fact, let me start writing. All right. So let's say carbon dioxide. Carbon dioxide. Carbon dioxide is, let's say, is larger than water. Okay, it's larger than water, but this is a big but. I mean, no matter fact, let me put that even in uppercase. That's the point. It's out. But, all right, that's not the proper way to write, but I'm just doing that, right? But water is less soluble. A matter of fact, I could say insoluble in oil, because remember now, the phospholipid, which is the lipid with the fatty acid ends, they themselves are non-polar. And so water cannot dissolve in oil substances. So therefore, they'll not be attracted to the molecules or the fatty acid part of the thing. All right, but but water is insoluble. Okay, insoluble in lipids. Okay, let's say in lipids. So and lipids and water do not mix at all. All right, so put lipids right here. In lipids, they do not mix at all. So just because water is smaller, it doesn't mean it's going to transport faster, diffuse faster through the membrane, for the sole reason because water is insoluble in lipids. All right, and next reason also that make carbon dioxide diffuse faster than water, carbon dioxide is gaseous. Okay, so carbon dioxide is a gas. I keep on doing this. Carbon dioxide is gaseous. Okay. All right, and gases diffuse faster than what? Liquid. Okay, so gases will diffuse faster than liquid. Okay, because again, remember, and this is based upon the kinetic energy of the molecules, because gases has greater kinetic energy compared to water, to compare to liquid, and so hence they move faster. All right, and so there's another reason, because carbon dioxide is non-polar. Carbon dioxide is, you can actually write that in as well. Carbon dioxide is non-polar. Water is polar. You see? And so for that reason, again, we know for sure that water will be less compared to carbon dioxide. So just make a note right here. Again, just to give you some more information here. All right, so carbon dioxide, is non-polar. Okay, it is non-polar. All right, while water is polar. All right, and again, polar substances do not do well with non-polar substances. And so remember now, because the phosphate ends on the outside, water will be attracted to the phosphate end, but in the middle or the central part of the membrane, you have the fatty acid ends or the fatty acid tails, which is going to be difficult for water to pass through the fatty acid because, again, they are non-polar. Okay, so polar substances do not attract to non-polar substances. All right, or do not dissolve in them. All right, so that's very important right here. Let's talk about glucose now a little bit. And let's say, because glucose will be next in line, and then you have RNA after that. So the reason now why glucose will be before RNA is simply because glucose is polar but is uncharged. RNA is polar uncharged. But let's even talk about that a little bit. In terms of complex, right? Glucose, I should say, glucose is more complex compared to carbon dioxide, and what is larger. They can do the relative molar mass. I know that glucose is larger. So in terms of size, water is the smallest one, but water will diffuse after carbon dioxide simply because of the polarity and the solubility. All right, glucose now is larger than water and carbon dioxide, but it's smaller than RNA. Okay, so glucose is larger than carbon dioxide, all right, and water. Okay, and water, but smaller than RNA, because RNA is a larger and a more complex structure. All right, what is also important is that in terms of the last two, glucose and RNA, glucose, right, is polar uncharged. It's polar and uncharged, which the overall structure is uncharged. So there's no so it's polar uncharged. All right, while RNA is polar, all right, polar charged. Let me just say polar charged. Let's put it like this, polar charged. And what I want to point out again, again, for two reasons, because RNA is larger and is also polar and charged. It therefore means, because again, the fatty acid ends, because they themselves are non-polar, they will not attract it to the polar charged substances. Easier for them to attract the polar and uncharged substances. Remember, glucose could dissolve in water, so glucose is polar but yet uncharged, right? That's something to point out as well. And so these are the reasons in terms of the order. All right, so let's jump to the next question here to see how fast we could finish with this one. And so the next part of this question now is that phospholipids are major components, right, of all cell membranes. With the aid of an annotated diagram, explain the organization of membrane lipids. It's a discuss the structure of cell membrane, specifying the organization of membrane lipids and the various ways in which proteins can associate with the membrane. All right, so when we talk about membrane lipids, what we actually talk about here is phospholipids, right? Just to bear that in your mind. All right, so the membrane lipids talk about phospholipids. That's what they're talking about. All right, so when we talk about that, we also want to discuss the structures. We're going to talk about the structure of the cell membrane. I want to specify the organization of the membrane lipids, so how the membrane lipids are organized. And then the last thing we're going to talk about is how proteins associated with the different ways in proteins associated with the membrane. So these are the things that we have to focus on. All right, so in the space, here's a draw diagram here. So I'm going to draw a diagram right there, and it's an annotated diagram. So let's think about the phospholipid bilayer right here. And actually giving it the word. This is the last question, in fact. It's an 8-mark question, right? And so what I'm going to do here is to draw the phospholipid bilayer structure. Let's do it right here so I can label on both sides. And so what you're going to have is some circles. Let me do all the circles together first, and then I'll draw my thing at the end. They're actually pretty close together. All right, that's enough. I will draw just only that. I don't need to go all the way through. All right, so I just need to draw a portion. All right, so each of them has 2 fatty acids. All right, and again, let me draw the lines here so it'll be easier for me. All right, as a matter of fact, let me draw the circles here, which is a phosphate. All right, so I'm not the greatest artist here, but I'm trying my very best to draw something. All right, so yep, so great there, awesome. All right, so what I'm gonna do now is to do some labelings, right? So let me put my label lines in blue so you see exactly what I'm gonna label. So we need to annotate the diagrams as well as annotate the diagram. So here I'm gonna label this part. I'm gonna label this part. All right, and I'm also going to talk about what these actually use for. All right, and so let me just kind of erase this line a little bit too long. All right. All right, so that part is good. All right, great. So let's do the labelings real quick. So annotation is really labeling and probably explain a few little things, state a little thing right there. So this is, again, you can label it as the phospholipid bilayer. So let's say phospholipid bilayer. Okay, so this is a phospholipid. All right. And this is a bilayer. All right, just to make a note right there. All right, so right here now, this is what they call the phosphate. So right here is the phosphate part of it. Okay, so there's a phosphate. And the phosphate here is what they call the hydrophilic head. Okay, so this is hydrophilic head. That's the hydrophilic head. And here now it is the fatty acid part of it. So this is the fatty acid. All right, so it's the fatty acid part of the structure. And this is the hydrophobic. So this is hydrophobic now. The hydrophobic tail. That we describe as the hydrophobic tail. And so the head part of it, since it's hydrophilic, it attract water. Just to make a note of that. So attract water. Oh. So attract water. Oh, come on. All right, so attract water. Okay, great. And then this part here now, which is the tail. No, okay, let me erase this line. I need to this one. I want this on the tail. All right, so erase that line, put it on the tail part. This is what I want to actually talk about, the tail, right? And so the tail now, it repels water. Okay. Just to make a note of that. All right, so you have repel water and attract water. So the head will attract water, the tail will repel water. All right, so that's to make a note of that. Again, is a phospholipid. The entire thing is called a phospholipid. So here, if you want to label this entire portion right here, all right, we could do this and say one of them is called a phospholipid. Okay, so this is a phospholipid. All right, so again, the entire thing is a phospholipid bilayer because there are two layers. All right, one on the outside, one on the inside of the membrane. All right, and they both form the membrane. All right, so here now, we can now talk about some number of different things. 8-mark question. So we're going to talk about a number of things. I'm going to give them in point format, a matter of fact, so just to make life easier. So I'm going to kind of just run through them real quick. So I could give you some 8 marks. I'm going to talk about all the protein associated with it. And so here now we can say that the head is polar, just to make a note of that. Head is polar. And since it's polar, it is hydrophilic, right? So it's polar / hydrophilic. All right. So because it's polar, so let's please, if you're writing this, make sure you write it the proper way. So the head is polar, and because it's polar, it's going to be hydrophilic. Because what is polar? So polar substance is attracted to the polar head, right? So just to make a note of that. So it is hydrophilic. Because it's polar, it is hydrophilic. And then the tail now, the tail is non-polar. And because it's non-polar, then it's going to be hydrophobic. Okay? So just to make a note of that as well. So because it is non-polar, it's hydrophobic. I'm just going to give you in points. So at least when you're writing and going through the exam, you know what to do. All right. And what I could also mention, because the phospholipid, since the phospholipid itself has hydrophilic and hydrophobic ends, they actually call amphipathic. Okay. So I could write it down right here. No, let me put it right here for you real quick. So fast I could do this. So because it's hydrophilic and hydrophobic at the same time, we can say this is what they call amphipathic. So this is amphipathic. All right. Just to make a mention of that. All right. So great. All right. So now what we can also talk about here, some other things. And so you can say the lipids. No, the lipids are arranged in a bilayer or a double layer, into a bilayer, which means double layer. Okay. As I mentioned, one inside and one on the outside is a double layer right here. All right. And then now the next thing to note is that the heads, just to talk about it, the heads, because he asked us to explain the arrangement of these things. So the heads now are on both sides. The heads are on both sides of the plasma membrane, right? Of the membrane. So one on the inside, one on the outside. So both sides, I'm going to say inside and outside. Okay, inside and outside. All right, so at least you know exactly what I'm talking about when I talk about the both sides. All right. So that's now important. That's very important right there, right? So just to clarify what I mean by both sides again, I'm just making a little note right here. So this notation I'm going to talk about right here is that one head or one set of heads, all right, or one layer. So one set of heads / layer, all right, is in the cytoplasm is in the cytoplasm. So you see one in the cytoplasm of the cell, while the other one, so let's say while the other is on the outermost part of facing, or while the other, let's say, is facing the extracellular fluid. So we have one that facing the cytoplasm, which is the internal fluid, all right? And so we have the other one is facing the extracellular fluid, is the fluid on the outside of the cell, all right, which is the tissue fluid. All right, just to make a note of that. Now, what we can now jump into to finish this up now is the arrangement of the proteins, or the proteins associated with the cell membrane or the plasma membrane. So let's talk about the proteins on this page right here. We finish up with this now. So let's talk about proteins. The proteins associated with the, all right, so protein associated with the membrane. So there are a number of ways that proteins associate with the membrane. And I'm going to kind of give you three, well, I could go up to four. But what I want to generalize first, right, is that there are two ways in which, generally, there are two ways in which proteins are associated with the membrane. One is that the protein may go straight through the phospholipid bilayer, and others will be peripheral. Okay? So those now that are transmembrane and those that are peripheral. So I'm going to put just put them right here just to kind of give a general idea in which their layout will be. So you have those that are called transmembrane protein, all right? And so transmembrane protein, the one that goes all the way through, and those that are what they call peripheral proteins, which are found on the surface of the protein. So those are the two general ways. All right, so again, proteins may be transmembrane proteins or peripheral proteins. All right, so now let's talk about now the first one. And so one way in which the proteins may be associated with the membrane is that they pass straight through. All right, they pass straight through. All right, and so these are what they call a trans protein. So transmembrane protein, the transmembrane. All right, so the transmembrane protein. All right, so these now, again, they go through the membrane or across the membrane. Okay, let's say proteins. All right, and say they now, they go through, all right, or across the membrane. All right, just make a note of that. All right, so when I draw the phospholipid bilayer, I could show you something on the side right here. So let's say this is a phospholipid bilayer right there. All right, so I'm going to do that in a small space. All right, and so I want to kind of have an idea exactly what I'm talking about. And it comes like this. And so what is happening now is that the protein will be going all the way through. So from the outer surface to the inner surface. So this is a protein that will go through. And these are normally what they call facilitated protein or channel proteins. Okay, so we have channel proteins. All right, so once you talk about cell membrane or plasma membrane, you may see the word channel protein or facilitated proteins and so on. All right, so that's the protein right there. And the phospholipid bilayer is right there. All right, let's go to another way right here. No, let's even finish with this. There's some more information I could give you on the transmembrane proteins that they go straight through the membrane, which is just one point to make. All right, that's just only one point to make right there. But what I want to also point out to you real quick right here is that the trans protein, they are generally what they call amphipathic. And they're amphipathic because one end will be hydrophilic, one will be hydrophobic. Okay, just to make a note of that. So they are truly, just to make a note on this, they are amphipathic. All right, because one end of them will be hydrophilic, one will be hydrophobic. All right, and also what I want to point out again, that they may arrange in different ways. So they can arrange in single helix, which means there's one helix line of protein going through. So they can be single helix, they can be single helix, they can be multiple helix, they can be multi-helix as well. And also they can be coiled, all right, or coiled. That's the way they may seen through the membrane. All right, just to mention that. All right, let's go to second type. All right, and we have those now that may be found within the cytoplasm. All right, and so I can call these now cytosolic or cytoplasmic monolayer proteins. So the second one right here, let's call them, let's put this number 2 right here. So a second way which you can associate with the membrane is by what they call cytosolic, all right, monolayer. And again, we say monolayer because a single layer on the inside. Or we may call it cytoplasmic. So cytoplasmic monolayer. Okay. All right, just to give it a name. And again, this is a peripheral. So the one I did first, number 1 is what they call the transmembrane protein. So we're talking about now peripheral proteins. And so one may be cytosolic monolayer or cytoplasmic monolayer. And this one is actually inside of the cytoplasm. So these are found on the inner layer in the cytoplasm. So so let me just make a note right here. So these proteins, let me just put proteins right here. All right, so these proteins now, let me get back in this line. All right, yep. All right, so these proteins, right, are found within the cytoplasm only, or found only in the cytoplasm. All right, okay, are found only in the cytoplasm. Okay. All right, so you only see them in the cytoplasm, so they connect to the membrane that is in the cytoplasm. So again, let me just do a quick drawing to demonstrate that I'm saying here. So if you have the phospholipid, right? That's the phospholipid right there. And so if, let's say inside here, the one that I'm drawing now, let's say those are the inside of the cell, which is the inner layer of the cell, then the protein structure may be right here. So this would be protein structure I'm scribbling right here now, attached to the internal part only. That's just called peripheral on the surface. All right, so let's go to another type. Here we'll also what they call the outer layer one, is called the plasma membrane monolayer. So this is the third one. We can call this one plasma membrane. The plasma membrane. All right. The plasma membrane monolayer. And the word monolayer just says a single layer, okay? And these proteins here, monolayer proteins. Now these proteins, are found only on the outer surface, right? So they only attach to the outer surface of the membrane itself. All right, so they're only on the outer surface, which they are in the extracellular fluid. All right, so let's say in the extracellular fluid. So that's where they are. So one is in the cytoplasm, and then one is in the extracellular fluid. All right. So, and again, the names are associated with them. So, what other name for this protein here that are found on the outside, just to make a note of that, they are called the extrinsic protein. Just to make a note of that, okay? Extrinsic proteins. All right, great. All right, and there is another one that I can talk about. Just to make, I'm just going to make reference to it really, and you have one of this called the lipid link. Okay, the lipid link, and the lipid link is another peripheral again, but this one is just linked to the membrane by a lipid. Okay, so just to make a note of that. So the lipid link type of protein. All right. All right. So this is where it is. Just to make reference to those. Those are the different ways that proteins can associate to it. You also have proteins that attach other proteins as well. So you have lipid link and you have protein link. Okay, so let's talk about protein link protein. That's one kind of a funny type of one. Protein linked proteins. All right, so protein linked proteins. So in other words, protein linked to proteins that connect to the phospholipid. Okay, so there are many ways in which you can find them linked to the membrane itself. All right, so great. So the last is not necessarily important in terms of discussing what we just discussed, because they are just examples of peripheral proteins, right? Just the way they link to the surface is either by lipid structure or another type of protein, or a specialized type of protein. All right. So this is where we're going to stop for today. Again, I'll do that at 2 questions as soon as I possibly can. So until that time, keep safe and be blessed.